Hello, students. Last time we met, we reviewed the difference between somatic, or body cells, and gametes, or sex cells. We saw that all of the somatic cells that make up our body are diploid cells, as they have two copies of every kind of chromosome, one from mom and one from dad. In contrast, we saw that gametes were haploid, with only one copy of every chromosome. Today, I'm going to first describe why it's important that the gametes are haploid, and then I'll talk about meiosis, which is the process that our bodies use to make haploid gametes. Alright, so to answer the question of why it is important to have gametes that are haploid instead of diploid, let's review, on a cellular level, the process by which a baby is made through sexual reproduction. Recall that in sexual reproduction, each parent must donate a sex cell, or gamete, and that these gametes are haploid, meaning that each has only half the number of chromosomes found in the organism's body or somatic cells. As you know, the female gamete is the egg, and I do want you to go ahead and draw along with me, please. We've got lots of pictures on this particular slide. This egg will have a total of 23 chromosomes in it because it is haploid. The male gamete is the sperm, and just like the egg, it will also have a total of just 23 chromosomes because it also is haploid. Now, as you know, in order to make a new organism through sexual reproduction, the sex cells from each parent have to join together. So here's our egg, and here comes our sperm, and they will join together in an event called fertilization. The new cell that is formed as a result of fertilization is called a zygote. This zygote is the very first body cell of a new human being. The reason that this is the first thing that we consider to be a body cell of a new human being is because unlike either the egg or the sperm by itself, the zygote is diploid, meaning that it has the full set of chromosomes that are needed to make a normal human somatic cell. So to recap, this cell seen here is a zygote. You all at one point were a zygote, the one cell organism that you started with. And because the zygote results as a product of the fusion of a haploid egg and a haploid sperm, each carrying 23 chromosomes, added together, this zygote has 46 chromosomes. And haploid plus haploid means that this cell is diploid. Okay, so now once we have our very first new human diploid body cell, the next thing that we have to do is make more of these cells. And the way that we will do this is through a method you've already studied, and that is using mitosis and cell division. Because the zygote is diploid, and it's going to be the pattern that we're going to use to make all new cells from here on out, means that each new somatic cell that we create is also going to be diploid. Remember, we made a big deal in the last section about how the end result of mitosis is two cells that are identical to the original cell. So if the original cell is diploid, if we're making more by mitosis, all of our subsequent cells will also be diploid. So we're going to diagram how this works. So we've got, here's our zygote, and what will happen is that this zygote will divide through mitosis. It's going to make two copies of the original cell, and so now we've gone from a zygote to a two-celled organism. We know that mitosis can continue. Each of these two cells can divide and form a 4-celled organism. And then continuing on, those 4 cells can divide to form an 8-celled organism. And so on and so on. And I've got many arrows here because this will actually continue over the course of 9 months. After about 9 months of adding new cells to this organism's body through mitosis and cell division, you will have enough cells to make this beautiful miracle baby. If you're wondering, you do not need to be able to draw a baby that well. You can just draw a little cartoon version of this baby. All right, so now that we've established why gametes are such special cells, because unlike all the other somatic cells in your body, gametes are haploid so that they can participate in reproduction. So the next question then that we need to address is, how do organisms make these special haploid cells, these haploid gametes? Well, we already know from our previous unit that new cells can be made through mitosis. But the catch here is that mitosis makes new body cells. And body cells are diploid. And so if what we want to do is make haploid cells, we can't use the same process that we used to make somatic cells. And so to make gametes, we have to use a new process. And that new process is called meiosis. All right, so meiosis, and I was supposed to have this animated to show up because I think this is blank in your PowerPoint, so you're going to have to write this in. Meiosis is, by definition, the process by which the number of chromosomes per cell is cut in half through a specific kind of cell division called a reduction division. To reduce something means to make a smaller quantity. So let's think, what quantity would it be that is becoming smaller through the process of meiosis? The answer is, we are reducing the number of chromosomes in each new cell made. We will start meiosis with a diploid cell, just like we start mitosis with a diploid cell. But by the end of meiosis, we are going to produce 4 haploid cells from that diploid cell. So actually, you can see that here. Here we've got, at the end, 4 cells. Oh boy. And if you compare the number of chromosomes in the nucleus of each of these 4 cells to the original, you see that each of the new cells has half the number that the original had. In contrast, if we think back to what we learned about mitosis in the last section, we said that mitosis is going to produce 2 new diploid cells from every diploid cell that it starts with. Because in mitosis, we wanted our daughter cells to be exactly alike in chromosome number to their original cell. But in meiosis, we don't want that. We want our new cells to be haploid because we're going to use those haploid cells to join together in fertilization. Okay, so that brings us to our next question. Why is it so important that these gametes are haploid instead of diploid? And the answer is that if the gametes were diploid, then you would have 2 diploid gametes joining together during fertilization. And if you add a diploid plus a diploid together, what you're going to get is something called a tetraploid. A tetraploid cell, that prefix tetra, I think I've gone over tetra before, but tetra means 4. That's what you get if you add a cell that has 2 copies of every chromosome to another cell that has 2 copies of every chromosome. So your zygote would be tetraploid instead of diploid like it's supposed to be. And so what's the problem with this? In most animal species, zygotes with too many chromosomes, like four copies of each is way too many chromosomes, they do not survive. So in order to make viable offspring, we do need to make sure that we have a way to make our gametes haploid instead of diploid. Oops. All right, so now we're going to dive into the details of how meiosis works. How meiosis works to make new haploid gametes. The large diagram on this slide shows an overview of all of the stages of meiosis. As you begin to investigate this diagram, the first thing that might occur to you might be something like, oh, this is just like the same as mitosis, except twice in a row. I want to right away clear that up. That is a misconception. For meiosis to produce haploid cells, what we're going to see is that the chromosomes actually have to line up and separate differently than they do in mitosis. So you might not be able to see these differences right away, but we're going to point out a couple spots where this occurs, where some differences occur. You've got one here and here. If you look at these stages, they may look like, at first, like metaphase and anaphase did when we looked at mitosis, but they're a little bit different. We'll see that our end products, of course, are going to be different with 4 cells that are haploid instead of 2. And so I'm going to go through each of the steps of meiosis on the subsequent slides here to really clarify the differences. And but I did want to point out that this slide is a good slide for you to refer back to, use as a reference when we're done going through the details to understand the big picture of how these steps all fit together. And this is also a good resource from which to study. Okay, so in order to understand the steps of meiosis, we actually first have to pause and take a look at some important vocabulary. So the next 2 slides are going to be a little bit of vocabulary, and then I'm going to use that vocabulary to explain what's going on in meiosis. So to begin with, we have this first term, which is homologous chromosomes. Homologous chromosomes are a pair of chromosomes that contain the same gene sequences, and each one of these pairs is derived from one of your parents. So one from mom. I'm sorry, I'm on my Mac. Oops. So my writing, that's why the writing is so bad, because I'm trying to do it on my Mac. So bear with me with the writing. But when I say one derived from each parent, it means one from mom and one from dad. Now, this term homologous, you've seen it before when we did our unit on evolution. In that unit, we called certain body parts between different organisms homologous if they were similar in their origin. So not identical, but they were similar. And so here, the term homologous still means that. It means 2 chromosomes that are similar. Okay, so if we're going to look down here in the diagram, these 2 chromosomes are an example of homologous chromosomes. Let's say that this one came from mom, and the other one here came from dad. And so you would find both these copies in your cells. And, sorry, that looks like bad. It's supposed to say dad. And the reason we call these homologous chromosomes is that at each, as you travel down the length of the chromosome, if, for instance, you have a gene here where I had that yellow dot, if you have a gene there for eye color, on the chromosome that came from your dad, you are going to have in that same spot also a gene for eye color. All right, so that spot is the gene for eye color. That means on the homologous chromosome, you have a gene for eye color. Okay, if you travel further down the chromosome and maybe you've got a gene for metabolism there, then on the homologous chromosome from mom, you're going to have a gene for metabolism. And so on all the way down the chromosomes. Okay, so it gets a little, for students, this sometimes gets a little more complicated because remember that when you're looking at the chromosomes like they are in the very left bottom picture, this would be during a time when the cell's not dividing. But remember, before the cell divides, it has to duplicate its chromosomes. And so what that means is you're actually going to have, oops, sorry. You do not have to make that blue blob I just made. I meant to do this. If this one's the one from your mom, then when you duplicate your chromosomes before cell division, you have the one from your mom and also a copy made during S phase. Okay, so that's the one from your mom, and then that's the copy of the one from your mom. And then the homolog from your dad, if this was the homolog from dad, then right before cell division happens and we duplicate our DNA, we also have to make a copy of dad's chromosomes. So at this point, if you've got a cell that is getting ready to go through cell division of some sort, you're going to have your original diploid numbers, your original two homologs, one from mom and one from dad, and then a copy of mom's and a copy of dad's. Okay. Additionally, I've got one more thing I want to mention about these homologous chromosomes, and it's that even though homologous chromosomes have the same genes in the same location, the actual sequence within those genes may be different. And so we call that difference in sequence that may occur, we call those different alleles. So alleles are different versions of a gene. So for example, I mentioned before that you might have a gene for eye color down here on mom's chromosome, and I shouldn't say mom's, the chromosome you got from mom, it's your chromosome, but the copy you got from mom and the copy you got from dad, I said the eye color gene is in the same spot. But if we go over here, I want you to notice that sometimes that eye color gene might be a sequence that gives you blue eyes, or it might be a sequence that gives you brown eyes. Okay, so the chromosome you got from mom may be carrying a blue version of that gene, while the chromosome from dad, still an eye color gene, but it may be carrying a brown version. These two different versions are called alleles. Okay, and so they don't always have to be different. For instance, here we see that maybe mom has a brown allele and dad has a brown allele. Or maybe mom and dad both gave you blue alleles. Okay, so the alleles can be the same, but they don't have to be. All right, so a little bit more vocabulary before we go through the stages of meiosis. We will shortly see that in the first prophase of meiosis, the duplicated homologous chromosomes, like you see here, so these are like the ones we just saw. We have maybe these ones were from mom, so it's mom, the copy that came from mom, and the duplicate the copy that came from dad and its duplicate. So we would call these homologous chromosomes with their duplicates. And so what will happen in the first prophase of meiosis is that these homologous chromosomes will come together to form a structure like this. This structure here is called a tetrad. It refers to the fact that you've got two replicated homologous chromosomes all stuck together. And so in total, that actually makes 1, 2, 3, 4 chromatids, all kind of associated with one another. And so we're going to call that a tetrad because remember, tetra is a prefix for 4. So this structure where you've got the two homologous chromosomes and their duplicates all pairing up together is called a tetrad. In the first prophase of meiosis, we're going to see that this tetrad formation is really important because it's what is needed in order for this next event to happen. And that event is called crossing over. Crossing over is a process in which homologous chromosomes will exchange portions of their chromatids. And so that's what we see in the bottom diagram. You'll notice you've got your homologous chromosomes. This A, B, and C refers to the alleles of the particular genes that are there. To start with, if you've got this chromosome, its duplicate is going to have the exact same alleles because you made an identical copy. But over here, on the other homologous chromosome, you'll notice same genes, but different alleles. That's why we wrote them in lowercase letters, to just indicate that same genes, but different alleles. So what will happen is that when these two homologous chromosomes and their duplicates come together to form the tetrad, you can see that down at the bottom, there are parts of the chromatids that are actually going to wrap themselves around each other, and they're going to switch some pieces of the chromosomes, so that at the end, if you keep on going here, you'll see that you've got actually 4 different chromatids. Now, you've got this original one, where you see capital A, capital B, capital C alleles. But because these crossed over, this second one actually has a different combination. It's got capital A, capital B, and then lowercase c. And this one as well, because of the crossing over event, has lowercase a, lowercase b like it had before, but now it's got an uppercase C allele that it got from the other chromatid. And then, of course, this last one has all its original alleles. So the point of this crossing over event is, hopefully you can tell by what we just did, you get 4 unique chromatids. All right, sorry, this is painfully slow. All right, so we are now ready to look at the stages of meiosis. So let's go ahead and erase this, and here we go. The first thing I want you to recognize is that unlike mitosis, because we have a different goal in meiosis, we are going to have a different number of stages. So meiosis actually consists of 2 phases. The first is called meiosis 1, and the second is called meiosis 2. So why does meiosis require 2 phases? This might be another one where I thought I animated it, but I didn't. I don't know if you have this written in. So if you don't, write this in. The reason meiosis requires 2 phases is because remember that typically in a cell cycle, when you're done, you still have 46 chromosomes. But if our end goal is to make gametes and gametes need to be haploid, then we're actually going to have to take that 46 and divide it again so that we can get 23 in each new cell that we make. Okay, so I'm going to go ahead and describe each stage of meiosis 1, and then on the next stage, describe each step of meiosis 2. I want you to kind of follow along using the pictures. All right, so first, just as in meiosis, there is an interphase before we actually start meiosis. Now, you don't see it on this picture, but it does exist. We would call this first interphase, interphase 1. And the same things are going to happen in interphase 1 as happen in interphase of mitosis. In particular, in interphase 1, we will replicate our DNA. All right, from there, we will move into prophase 1. Prophase 1 will have many similar events going on, just like prophase does in mitosis, except 2 events that are unique to prophase 1 that we don't see in mitosis, and that is the formation of a tetrad. So those homologous chromosomes with their duplicates are going to come together and form that structure we looked at on the last slide where you have 4 total chromatids. And remember, we said that was important because the formation of the tetrad is going to lead to that crossing over event. And with that crossing over event, you're going to end up with 4 unique chromatids. All right, next the cell is going to move into metaphase. Okay, so we had prophase 1, we're moving into metaphase 1. And if you look at metaphase 1, at first it is going to look like metaphase in mitosis. But what I hope you notice is that in contrast to metaphase in mitosis, in metaphase 1 of meiosis, our chromosomes are lining up in homologous pairs. So what you should see here is actually 2 lines of chromosomes, a line here and a line here, instead of one single file line. So the reason why this is important is because it's going to affect what happens in the next stage, which is anaphase 1. If we were looking at mitosis, we would know that in anaphase of mitosis, the sister chromatids split. But in this case, because they lined up originally in homologous pairs instead of in a straight line, what's going to happen in anaphase is that the sister chromatids will remain attached to each other. And it's the homologous chromosomes that are going to separate. Okay, so you can see here, you've got one chromosome plus its duplicate, and they are still attached to each other as they are being dragged to either side of the cell. So in anaphase 1, the homologous pairs will separate and move to the poles. Then finally, you're going to get to telophase 1, and this should look the same with this. These events should look the same as do telophase and cytokinesis in mitosis. The only difference, which I didn't write down here, but you should be able to note, is these chromosomes are still have their sister chromatids attached. All right, so from here, these 2 cells are going to move in to the next stage, which is meiosis 2. Okay, so remember that when we are making gametes, we want them to be haploid. So both of the cells, and that's why you're going to see a set of 2 cells in this diagram, is because the first phase of meiosis made 2 cells. So now each of these cells, in order to get themselves down to a haploid number, have to be divided again. And so what will happen is and it's not shown here, and so I apologize, but just like before we started Meiosis 1, there was an interphase. In Meiosis 2, it starts again with an interphase, and we'll call it interphase 2. But, and this is a really important but, in interphase 2, the DNA is not replicated. Okay, so remember, normally we replicate our DNA so that we have the same number of chromosomes at the end that we do at the beginning. But here, we actually don't want the same number of chromosomes at the end. We want to cut down or reduce the number of chromosomes. So we're not going to replicate our DNA before we do this set of steps. And then you can kind of look through them, and as you look at prophase and metaphase, you can see now these start to look like they did in mitosis. You've got a single file line here, a single file line here. You have chromatids separating in anaphase. And then finally, at the end, in telophase and cytokinesis, the events look the same, but remember, the total outcome is different. At the end, you have 4 cells because this cell divided twice, so you're going to get 1, 2, but then we've got to count these ones down here as well, so 3, 4 cells at the end. And not only do you have 4 cells, but because you didn't duplicate your DNA at the beginning of this, your 4 cells are haploid. That means that if indeed we're talking about human cells here, each of these cells will have 23 chromosomes each. And I'll just abbreviate that because my writing is slow. All right. And so that was the goal, and meiosis accomplishes it. Now these, you'll have to remember that these daughter cells that are formed, they're not you can't find these special daughter cells anywhere in your body. This whole process only occurs in the reproductive organs. So it's not as if your skin cells are ever going to be going through meiosis. So the reason I point this out is these 4 cells will further develop into the shape of a sperm or the shape of an egg. And you can actually see that on the very first picture of the title slide. You can kind of see how those structures are developed at the end of mitosis. I'm sorry, at the end of meiosis. Okay, we are done for today. Thank you so much for sticking with it through this long lecture. You can check Veracross for any additional instructions about what you should do now. And we will go over this again tomorrow so that I can clear up any misunderstandings. And so I look forward to seeing you then.